Good afternoon, friends. It's a Friday afternoon and it's going to be 3. And we are back from the Scientific Society of Aarupadai Veedu Medical College, a constituent unit of Vinayaka Mission Research Foundation, with our next edition of the Forefront of Future Frontiers webinar series. As you know, we started this endeavor during this COVID pandemic peak times in the month of May, and we have actually completed 3 series. On series 1, we started with what are the recent advances across various fields, ranging from ophthalmology to cochlear implant to cardiac myopathy, etc. Then we moved on to explore how COVID has impacted our life, and as humanity, how it has affected us. So that is what we did through a series of 4 talks from humanity's response to COVID, how to learn to cope up to live with COVID. Then we moved on to how institutions and individuals have put up resistance for COVID. So what can be done to combat this COVID pandemic and get back to normalcy? So we explored various aspects in that series 3 on that particular thing. So we talked about how education can be restored back to its feet by online remote learning. We spoke about how COVID can learn lessons from climate change aspects. We spoke about how anesthetists and surgeons can come back to normal, and the perioperative care of patients can be optimized. We spoke about clinical trials in COVID in the last week's edition, and what is the science, what is the ethics, and what is the actual practice which is going on in COVID. So that was our series 3. Now we are moving on to series 4, and the theme for series 4 is to explore what are the future frontiers in research. Okay, what is new in research and how these things can affect our clinical practice. So we will be exploring various aspects of research which can have a lot of impact on our day-to-day clinical practice. So series 4 FFF for you, and we have a very interesting talk today by Dr. Madhu Lal, who is our Dr. Vijayalakshmi's daughter. She's very happily seeing. So we have she's going to talk to us about translational medicine and how it can help in advancing clinical practice. She's going to use COVID diagnostic testing as a case study and then explore these aspects. So before we move on to the talk, I would request our Dean sir to welcome the speaker and say a few words. Over to you, sir, Dr. Kotur.
Yeah, thank you, Professor Mahalakshmi. Indeed, as head of the institution, and I am very happy to welcome our speaker. And this is the 14th series we started on 25th May. Platform, the various efforts from different fields have shared their expertise, their knowledge and experience, and certainly the number, of the beneficiaries. While going to the report of the webinars Mahalakshmi shared today, it is really we have reached the 4-figure, the number of beneficiaries by this platform that has been created by the Scientific Society with the patronage of our Honorable Chancellor, Madam Chancellor, and the Board of Management. Certainly, it's a wonderful contribution on the part of our small institution for the medical fraternity. And in this platform, a lot of exchanges have been happening, and majority of our lectures have been related to the current pandemic that is going on, and we have learned to live with it. And though translational medicine, though I say is not that new concept, we all know that it is a bridge between the science and the practice. It links the laboratory with the patient care, patient findings. And now in national emergency, in a global emergency like this, health emergency like this COVID-19 pandemic, the process of turning the research findings or the observations which have to be translated into patient care for the benefit of the mankind have to be faster than ever. And we all know that the COVID-19 itself has posed so many challenges from all fronts, on all facets. And under such circumstances, see the role of this translation medicine or translation research for overall benefit of the mankind is quite important. Be it developing a treatment or an intervention, or be it testing the efficacy or effectiveness of any test, or be it the dissemination and implementation of research to avoid system change. We learned about a system thinking, system change has to be brought in. So these some thoughts which come to my mind. I'm sure our present speaker, Dr. Madhu Lal, will be sharing her experience. And she is from the other continent. We had all, we had from Southeast Asian continent, we had Australian continent. Now it is a American continent. A wonderful thing. And so I'll say our FFF webinar has become really international. And Mahalakshmi must be congratulated. You'll be very happy to hear these words. So with these few thoughts, over to you, Professor Mahalakshmi, to introduce our speaker today.
Thank you very much, sir. One small thing. So far, we have finished 19, sir. This is the 20th in the FFF webinar series. The series 1 had 6 talks. Series 2 had 4. Series 3 had another 5. So this is the 20th. And we have Dr. Madhu. Yeah. Okay. Let me introduce Dr. Madhu. Her CV makes a very interesting reading. She has traveled really places. She started with her MSc in Masters in Biochemistry from the University of Mumbai in Madras, in India. Then she moved on to PhD in Molecular and Cellular Oncology from George Washington University. She's got a Master's in Bioscience Business from Keck Graduate Institute in California. She served as the director of NIH. I would request all the participants to please mute themselves. Sorry. And she has served as the director of NIH RNAi facilities. Madam, many of these things are kind of like Greek and Latin to people in practicing. She's been the program director for the Research Governance Council, and she has moved on to FDA recently. And her main interest lies in bridging the gap between academics and the translational aspects of cutting edge science and oncology. As our Dean sir aptly pointed out, that translational medicine is the what we actually call from bench to the bedside. So that is what she is. And she has developed a lot of models for safety and efficacy in drug developments and evaluation, especially with related to cancer chemotherapy. She is also, her team has come up with one functional genomics robotic platform to run high throughput phenotypic screens, especially when it is related to the human genome project. I think we'll be hearing about it a little bit more in the next thing, where we are going to talk about genomics also. And she's also done a set of standards for best fit models that can be used to answer specific questions as they pertain to disease biology in more complex in vitro cellular systems. So these are in short, which I got out of her CV. Maybe I have missed out a few things. Dr. Madhu herself can elaborate on these things as we go. This is our speaker in short for you. Over to you, Dr. Madhu. You can go ahead and start your talk. I've made you into the presenter, so you should be able to share slides now. Over to you.
It's still not unmuted.
Dr. Madhu, can you please unmute yourself? I've unmuted you. Please go ahead. Yeah.
Okay, perfect. Okay, thank you for that. Well, Dr. Mahalakshmi, thank you, and the dean, thank you so much for that extremely kind introduction. It's such a pleasure for me to be able to talk to you today about the role of translational science and medicine in the development of physiologically relevant disease platforms. It's something that's very near and dear to my heart. I'm trained as a molecular oncologist, so a lot of this work in terms of de-siloing clinical research and basic research has been done in the field of oncology, because truly we have not really still solved the problem of predictive models and, modeling human disease in alternative hosts to be able to develop effective and safe therapeutic options. But what I'll be talking about today is the extrapolation of that idea as it relates to infectious diseases, and sort of puts in perspective how we've had to modify drug development to deal with this pandemic. So a couple things that I do want to say. As I mentioned, the context of this talk is really modification of the drug development, of the therapeutic development platforms to deal with a public health emergency that's affecting the whole world. What it will not be is a comprehensive, didactic sort of list of every sort of therapeutic option available right now, because I'm sure you have access to that. The other thing is, I'm happy, outside of infectious diseases, I'm happy to serve as a resource for anybody who wants to talk about personalized medicine, especially in the context of solid tumors and hematological malignancies, because I really do think that we're coming to a place where basic researchers or the clinical researchers will work together within the spectrum of translational medicine to bring safer and more efficacious therapeutic options to patients faster. So that being said, I do have to start with a conflict of interest statement. The views presented here are purely my own and do not reflect the views of the United States Food and Drug Administration. As I'm sure you're well aware, most of the regulatory agencies in both in the United States, Europe, and Asia are doing their very best in terms of reviewing applications and sort of in an expedited manner. But this the detailed workings of how this process is not necessarily purview for public consumption right now. So that being said, I will start by talking to you about what translational science really is. And in the spectrum of research, I think we tend to come up with terms that are, that are new and exciting. But what translational science really means to me is being able to de-silo basic research and clinical research. And one would think that would have been intuitive from the get-go, but somewhere along the line, there was a divergence of research in wet lab research, that sort of focused on mechanisms and phenotypes and disease biology, and that sort of went further and further away from the patient itself. So what translational science does is basically it's just a mechanism of looking at disease, but in a plate. So it's bringing the patient back to the beginning of the research process rather than having the patient at the end of the research process where they're just the recipient of whatever therapeutic option has been developed. And again, like I said, this seems like it would have been an intuitive idea, but along the course of research, there was such a divergence in basic research and clinical research that a lot of therapeutic development sort of failed. There was a huge attrition of things that were potentially efficacious in human beings, because there was we had sort of not, we had lost the perspective of keeping the patient in the loop. So what translational science does is it focuses on both the scientific and the operational principles underlying each step of the translational process. So whether that's clinical research, whether that's the implementation of the findings of clinical research, it keeps public health perspectives in mind, preclinical research and basic research. And the idea really is, how do we all work together in the same spectrum to tackle the same disease, to develop new approaches, demonstrate their usefulness in human beings and disseminate the findings. So translational research is based on the fact that in the whole sort of continuum of drug development, we have not made too much of an effort to really develop predictive models for any disease. And again, like I said at the onset, a lot of this is based on my experience with solid tumors and hematological malignancies, but the same is true of of rare and orphan diseases, of infectious diseases. We still use cells in a monolayer to mimic human disease where you have systemic interactions between the organs. So that has obviously led to a huge attrition in the number of preclinical candidates that can actually progress towards clinical trials. And not only does this affect human health because we are unable to get better drugs to people faster, but it also is a huge drain on resources and the economy. So what does that where does that put us in terms of how we want to think about drug development? Well, that actually starts with beginning to understand what comprises the process of drug development. It's a multi-tiered process. It takes up to 12 to 15 years and can cost, depending on where you are, it is an extremely expensive process. And I cannot stress enough that the amount of time that one actually spends in research and development here at the beginning of this process is extremely important. Having robust research and discovery processes where you're able to identify a target and validate it and really have a high degree of confidence before it goes into the preclinical and the clinical stage is extremely important. And putting in that time and effort in this particular space to be able to really mimic the patient disease in whatever in vitro systems we have, to not use murine models just because they're available, but to be able to develop alternatives to animal models that more efficiently recapitulate the disease are extremely important. Now, while I say all of this in terms of, putting in this time, putting in this effort in research and development, what happens in the context of a pandemic like we have right now? We have a public health emergency. We don't have the time. We don't have the luxury of time to actually spend all this energy in research and development. So what it necessitates really is looking at the current therapeutic development process and then seeing what we can take out of it, sort of in a quick and dirty manner to be able to treat, in this case, people who are suffering from or have been infected by the SARS-CoV-2 virus. So what I'm going to talk to you today in the next couple slides before we jump into the pathophysiology of COVID-19 as it pertains to infection is high throughput screening, how that has really changed the way that we're able to repurpose drugs and the context of that with COVID-19 infection. So what I'm going to start off with is what a normal high throughput screening process looks like, and then from that perspective, knowing sort of the molecular entities and the chemical entities, the biological space that they act in, how are we able to repurpose drugs for specific targets, for specific pathologies, for specific phenotypes, and for patients with underlying morbidities. So in terms of looking at the technology for assessing the risk of drugs, now for the longest time, the drug development was looked at safety and efficacy, in silo. Again, not that that makes too much sense, because if you have something that works very well against the disease, but that is not safe in human beings, we can't actually sort of move that process forward. So if you compare the 1930s in terms of, where we are with respect to technology and everything that we've overcome, you can see great advances being made in many, many areas that affect our lives. But when it actually comes to how we're modeling human disease and how we're modeling safety in human beings, we really haven't changed the needle very much. So this really necessitates a paradigm shift in the way that we think about developing models for drug development. So under normal circumstances, what does the drug development paradigm look like? It constitutes identifying a target. And for the most part, we tend to think of targets as a protein, a gene, something that we can develop a drug against. But it really depends on the disease. Your target can be a phenotype. Your target can be a metabolic pathway. Your target can be metastasis. It really just depends on what you're trying to develop and the patient population that you're actually sort of going after. Because, for example, with hematological malignancies, everything is very mutationally driven, and you can actually identify genomic targets that you can develop drugs against. However, when you talk about solid tumors, especially of epithelial origin, you're looking at about 92% of the patient population that doesn't have a driver mutation. So you actually have to go after pathology in those cases to identify a target. So we identify a target and we create an experimental system commonly known as an assay. And then you sort of build in complexity into that assay that to the best of its ability can mimic the microcellular environment of that particular organ in the human being. And typically in high throughput screening during the discovery phase, we can test up to 100,000 small molecules. Again, I use during the course of this talk, I'm using the term small molecules loosely. It also encompasses biologics, generics, and anything that's a therapeutic option. You then make modifications to active chemicals to make them suitable for human use, which is a lot of medicinal chemistry, and then we test in animal models for efficacy and then go into clinical trials for safety and effectiveness. However, if you think about it, in the context of this pandemic, we don't have time. We don't have the luxury of money. We don't have the luxury of time to actually spend in terms of that whole drug development paradigm that I just described. So how does one actually at that point think about what we could be repurposing? So drug repurposing has been a tenet of translational medicine and high throughput screening for many, many years now, but it has only been in the face of not only COVID, but even with the Zika epidemic that we had a few years ago, drug repurposing really came to the fore because again, with Zika, we were having the populations that were affected was predominantly maternal, and it was an extremely insidious infection. Again, no time to actually develop an effective therapeutic, so we relied heavily on drug repurposing. And what drug repurposing really is we know the profiles of all the approved drugs in terms of safety. So all of the safety work that has to be done in human beings has already been done. We know what the safety profiles for those particular drugs are. But we know what the efficacy is against the particular disease that it was developed for. But what we have not done is tested it against different diseases. Now, over time and with the advent of high throughput screening, what has happened is we've been able to develop look at drugs in terms of the biological context that they hit. So now we are able to understand, for a particular drug, if it's used as treatment for disease X, what are the pathways or what are the pathologies for disease X that it's hitting? And then if these pathologies are similar to disease Y, can that same drug be used in the treatment of disease Y, which is exactly what we're doing with all the antivirals for COVID-19. Now, in terms of what encompasses drug repurposing, this is a very involved slide, so I'm not going to go through all of it. But the point that I want to make here is that, again, translational science is not just biology or medicine or oncology. It is the coming together of many in silico approaches, many in vitro approaches, many in vivo approaches to really try and understand what the molecular target is or what the physiological target is through molecular docking, looking at genetic associations, looking at pathway associations, and then looking at retrospective clinical analysis using real-world data and clinical trial data, and then taking all of that knowledge and then integrating it with actual wet lab research where we're doing this phenotypic screening, where we have a model that to the best of our ability recapitulates the disease phenotype, and then we're able to see whether the predictions from all of the computational approaches actually apply in relieving that particular pathology or phenotype for the particular disease that we're talking about. So what high-throughput screening really involves is a sort of a multi-pronged process, and as I mentioned to you before, a lot of it is automated, which sort of gives us a lot of confidence in the data that's generated. And I'll talk a little bit about the importance of having well-curated, well-normalized data, especially when you deal with patient data later on in the talk. But from a very high-level perspective, high-throughput screening involves assay optimization, screening of pilot libraries, large-scale screening, choosing the compounds that you want to sort of take into a preclinical space, and then following up with much more physiologically relevant, human disease-relevant assays. And there are a few centers here within the United States, one of them being the National Center for Advancing Translational Sciences. The other one is the Scripps Research Institute, which actually did a lot of the work that ended up, that I'll be talking about in a couple slides later, that ended up in developing candidates for refocusing for COVID-19 that have based all of their work on quantitative high-throughput screening. And what quantitative high-throughput screening really is nothing but looking at the same drug in the same experimental system over multiple doses, so that, you know, going into a preclinical space, it gives us a better idea of the extrapolation for the pharmacokinetics within an actual patient, so we're able to see what the range of dosage would be for a drug in an in vivo model, and then sort of predict on that for a patient. This is just a compendium of the different sort of libraries that are available for everybody to screen. As I mentioned to you before, when it comes to small molecules, they can be structurally, chemically very diverse, but they can also hit a very diverse biological space. And we sort of need a balance of both the chemical chemotype diversity as well as the biological, physiological space that it's being able to target to be able to develop an effective and safe therapeutic. I think I can't stress this enough. Again, like I mentioned at the beginning of my talk, you know, for the most part, for the last 80 years, we've been using two-dimensional assays to mimic human disease. It comes back to keeping the patient in mind throughout the drug discovery process. So it basically builds in physiological relevance. It builds in blood vessels into your 3D systems. It builds in sort of stromal environment, the inflammatory environment that you would actually encounter within an organ, and then being able to test these sort of medicines for efficacy and safety within the milieu of those organs. So we really have moved into a much more advanced cell-based model, so that, again, the confidence that you have in any therapeutic that you're developing is much higher as you move towards clinical trials. So to really wrap up that perspective, and that's sort of the Cliff Notes version of what translational medicine really is, it is an effort to bring applied science within the realm of basic research and clinical research, so that everyone is working together to get from bench to bedside. But what it really means is keeping the bedside at the bench from the get-go. So whether you're talking about assay development of biology through the screening platform, it requires a lot of medicinal chemistry optimization. It involves a lot of in silico methods. The idea really is that different disciplines come together to be able to develop a therapeutic that will be very efficacious in a human being, and being able to sort of hit disease that's mimicked by alternative animal models for treatment within human beings. So that being said, having given you the context of what modified drug development will look like in the event of a pandemic, what I'd like to segue into is what we are all facing now, which is absolutely sort of an unprecedented situation. With COVID-19 infection, even our infectious disease experts, led by Dr. Fauci, are the first to admit that we don't really know a whole lot about the infection. Interestingly enough, for many viral families, and we keep talking about coronavirus right now, but when we had the Zika epidemic, we were all talking about flaviviridase. With most viral families, the interesting thing is the evolution of the virus in terms of its ability to infect. And the other interesting thing is the host response. What is the host response? What are the comorbidities associated with the host response that actually makes someone more resistant or more susceptible to a particular infection? And I think that what infectious diseases experts sort of agree upon across the board is we have sort of undermined the host response in a lot of how we respond to an epidemic or a pandemic, but it plays a very, very important role. And what I'd like to talk about here really is in the context of this drug repurposing, in the context of developing assays in this sort of protracted time period that we have, what is the host pathogen relationship? How do we go from looking at this disease and the pathology, both pulmonary and extrapulmonary in a human being, to mimicking it at a molecular cellular level to be able to, in a very, very short time, turn around therapeutics that would be helpful to a patient population that is suffering from COVID-19. So as I'm sure you are no stranger to the human system and physiology. I know that to this particular audience, I don't need to delve into too much detail, but from a very 50,000-foot perspective, we're all aware of the pulmonary implications as of one of the first things that was seen clinically in many patients who were infected and died of the disease. However, one of the interesting things about COVID-19 that we're finding out now has been all of the extrapulmonary sort of manifestations of this disease in people who are actually recovering from COVID. And I'll talk a little bit about this in a couple more slides, but this is an extremely important area for clinicians because understanding the extrapulmonary complications of COVID and how to treat patients who have been infected with COVID and have recovered, but now might have different predispositions to other diseases becomes very, very important. And especially with people who have hypertension, who have cardiomyopathy, who have cancer, post COVID-19 infection, their disease, the disease might look very different. The disease manifestation might look very different. And understanding these extrapulmonary complications and what it has done to the different organs of the body will be extremely important in the next phase of patient and clinical management, not only for people suffering from COVID-19, but also from people suffering from other chronic conditions. As I mentioned to you before, for this particular audience, this needs no explanation, but there, not only from post-mortem analysis, but also from people who have recovered, there is a huge neurologic component to sort of infection with COVID-19. In addition, unsurprisingly, really, because of the ACE2 expression, which is a target for the SARS-CoV-2 virus, we have a lot of renal pathology associated with it. There is hepatic pathology with elevated aminotransferases, many gastrointestinal issues due to the ACE2 receptor being expressed in gastrointestinal tissues. One of the biggest ways that COVID-19 has this huge inflammatory profile is due to the triggering thromboembolism. We are all aware of the cardiac implications of the cardiac sort of manifestations of COVID-19 infection, not only during infection, but also post-recovery. It also has endocrine implications as well as dermatological implications as well. So when, if we think about everything that we went through in terms of the extrapulmonary complications of COVID, with respect to the host, why do we see this happen? Why do we see it being manifested in so many different organs? Is it a systemic response, or is it a more organ-specific response? And if you take a look at this, diagrammatic representation here, you can tell that, depending on where you see high, intermediate, and low ACE2 expression, you can actually sort of correlate that or extrapolate that with the physical and pathological manifestations that you have. So for example, with a lot of the cell types that I found within the gastrointestinal tract, there's a high expression of ACE2 expression, as well as within the liver, on the other hand, it's only found in cholangiocytes. Within the heart, we do know that ACE2 is increased in patients with heart failure, and within the lungs itself, again, going back to patient profile, host profile, what is the comorbidities, we know that ACE2 expression is much higher in smokers. The severity of pre-existing lung disease also is a huge sort of a pre-existing factor in how one will respond to being infected with COVID-19. Within the kidneys itself, ACE2 is widely expressed. We already know that this sort of widespread dysregulation of the renin, angiotensin, and aldosterone pathway by COVID-19 binding to ACE receptors is one of the major reasons we have the pathophysiology associated with the renal system, as the cardiorenal system in human beings, which in most cases leads to a high level of mortality. So, having talked to you a little bit about how the host responds and what the predisposition of a host or a patient is to viral infection, I'm going to shift a little bit to talking to you about the coronavirus family as a whole. This will not be an in-depth immunology discussion, but excuse me, I just want to give you a high-level overview of the mechanisms that are being targeted, the role of the inflammation response in this disease, and how drug repurposing sort of plays an important role in that. So when it comes to looking at the coronavirus family, whether you're looking at the SARS-CoV-1 or the MERS-CoV virus, one of the biggest we know that it sort of enters the cell through looking at the ACE2 receptors. But the host response to any virus usually is by being able to identify the single strand of RNA, that these are all single-stranded RNA viruses. But what the virus does to circumvent this is it sort of converts itself to a double-stranded version of RNA, which the host now recognizes as self and will not attack it. So if it's able to get away with doing this, and this is a very involved pathway, what happens is that the human host cannot mount an interferon 1 and 3 response, which is the most sort of prolific of the antiviral responses. Instead, what happens is you have now the secretion of many pro-inflammatory cytokines like interleukin-1, interleukin-6, interleukin-18, which in turn now become responsible for that huge cytokine storm, which is such a marked clinical manifestation of people who actually have severe COVID-19 infection. Interestingly enough, we see the same cytokine storm in many other viral diseases. Many very virulent strains of influenza also sort of elicit the same pro-inflammatory response. So what it ends up being is this delicate balance between an antiviral response and a pro-inflammatory response. And the patient's underlying sort of comorbidities play a huge role in sort of which one will be, which one will sort of win in a sense. The other thing about pro-inflammatory responses is much like the way solid tumors manifest, early on in the infection process or early on in the disease process, it tends to be protective because you are recognizing a non-self antigen. But much later on, when the disease has progressed beyond a certain stage, it becomes, this whole cytokine storm becomes very, very detrimental to the host, to the patient. In terms of the human host immune response for SARS-CoV-2, again, a lot of what you're seeing on this slide here has been, is what we discussed in the previous slide. But what I want to point out on this slide is this interferon response and the cytokine storm that we were talking about. It has an effect on all the different types of inflammatory cells, which in turn are what give you that cytokine storm and sort of increase patient morbidity. And in terms of what the cytokine storm actually encompasses, these are sort of the markers that we're looking at from the drug repurposing perspective to be able to dampen down so that we can modulate the cytokine storm response and the inflammatory response, which actually helps in better patient management. In terms of the first stage responders for a lot of viral diseases, it's the myeloid cells and it's the natural killer cells. But what ends up happening is that when you have this huge cytokine storm being released, the myeloid cell population as well as the natural killer cell population is extremely diminished in the peripheral blood supply, as a result of which they're not able to contain this pro-inflammatory response. So how does COVID-19 really, give us that sort of multi-organ failure? And what are the opportunities for therapeutic intervention? So we know already that, it enters through the ACE2 receptor and can cause direct lysis of the cell, which in turn, leads to cell death or pyroptosis. One of this widespread sort of pathology that we see in a lot of patients is due to disruption of the renin-angiotensin and aldosterone axis within the human being, which leads to many cardiac and renal implications. The direct sort of hit on endothelial cells not only affects the vascular system and sort of thrombolysis, but it also affects inflammation incredibly, thereby sort of contributing to this sort of multi-organ failure that we see within patients. And almost each and every one of these is associated with a dysregulated immune response because the host is no longer able to identify the virus as non-self. Okay, there we go. So how, in terms of a pathogenicity profile, how does SARS-CoV-2 compare with its close relatives that we're aware of, which is the CoV-1 and the MERS-CoV virus? Now, as you can see in this schematic, what is very important with and different between SARS-CoV-2 and the others is this organotropism. As you can see, for both of the other sort of close relatives, there was the infection tended to be contained due to tropism to a particular organ or tissue type within the body. But what happens with SARS-CoV-2 is due to its replicative ability and the ability to hide from host innate immune responses, there is absolutely no tropism. It's able to sort of invade into many organs, as we've discussed previously, and sort of cause that multi-organ systemic shock. And what you see here on the lower right-hand corner is sort of the immune response over years of post-infection. Again, we don't have the data for COVID-19 patients over that period of time, but as you can see, from what we can tell, at least right now, there tends to we tend to plateau out at a certain level of immunity. So the sort of communal knowledge or thought right now is that, post-infection, outside of the complications that you have physiologically, at least immunologically, there is reason to believe that we should be protected. And having talked to you now about, the host pathogen response, I just wanted to spend a little bit of time before we go into the drug repurposing, looking at the different cellular effects that the drug repurposing campaign can actually target. So as I mentioned to you earlier on, it doesn't always, when we think of a target for therapeutic intervention, it doesn't always mean that we're looking at a particular gene or a particular protein. It could be a particular process that we're actually trying to target. So, for example, here, you know, with a lot of the antivirals, we're trying to really sort of we're trying to inhibit genomic replication, subgenomic replication, and then really being able to attack viral structure. A lot has been said about chloroquine and hydroxychloroquine. I won't delve into that from the regulatory perspective, but I do want to say here that the idea here was not to go after anti-malarials as such, but to go after a particular process that chloroquine and hydroxychloroquine were targeting, which was basically to disrupt endosome maturation and viral budding. Now, why is endosome maturation and viral budding important? It's because this is the way that the virus can actually keep its lytic cycle going on within the cell, replicate, and then infect other cells. This one particular function of this one particular set of antimalarials was why it was thought that this could be effective in a particular subgroup of patients, but it did not necessarily necessitate the thought process that we should be using antimalarials, you know, across the board for COVID-19 infection. And then again, you know, we talked about the role of natural killer cells and cells of myeloid origin in terms of being able to really target this disease. Most severe COVID-19 patients present, for the most part, with very, very severe leukopenia. So the idea really is, are there antagonists and agonists from a therapeutic perspective that can actually dampen down that pro-inflammatory response of interleukin 1, 6, and 18, tumor necrosis factor as well, and then be sort of bring up the natural killer cell population so that we're actually able to combat the disease. And again, as I mentioned today, while I was talking about small molecules, there is a lot of work being done in terms of using neutralizing antibodies from convalescent patients, people who are actually recovering from COVID-19, and seeing what kind of an immune response that actually boosts for the patient. So having sort of gone through now the context of a modified drug development program in the, you know, when you're faced with an epidemic or pandemic, then having explored what the host pathogen relationship is, how the disease presents itself, and the opportunities for intervention by exploring what the host pathogen relationship is, I'd now like to sort of end my conversation where I began, which was with high throughput screening and the whole concept of drug repurposing. And one of the initiatives that was actually put together by the Scripps Research Institute, one of the institutes within the United States that does high throughput screening, using their small molecule library called the Reframe library. Now, the Reframe library is their signature library, and as I've shown you before, NCAS had, you know, a couple libraries that had diversity with respect to chemotype and biological space. So does the Reframe library in terms of the stage of development for all of the approved drugs within this library. As you can see, we have many drugs that are in phase 1, unclaimed or sort of investigational drugs, those that are in preclinical. A majority of this library are actually approved drugs. And this is the area that we really want to target because, as I mentioned to you before, for a lot of the approved drugs or for a lot of drugs that are in phase 2 or phase 3, we already know the safety profile. We already know the toxicity profile. So these are things that can be mobilized to be, you know, go into the patient population almost immediately because there is no need to actually have a safety profile. As I'm sure you're well aware, you know, and I think Dr. Mahalakshmi sort of bespoke to the clinical trials that are ongoing right now. But again, even with all the information available, we're not really conducting clinical trials you know, in the way that they're normally conducted. You know, they aren't necessarily blinded. There's a huge ethical issue with even including a placebo arm when people are so sick. So being able to leverage approved drugs or drugs for which we know a safety profile, and then test that, in cellular systems that mimic the disease is extremely important at this time. And in terms of the Reframe library and sort of the disease annotations that are associated with this, you see that there is a huge number that actually target respiratory pathology. A lot of them target the central nervous system. There's a reasonable proportion of anti-infectives, and of course, there, as with most libraries, a lot of them tend to be very oncology heavy. But this is a very, very comprehensive library of about 12,000 approved drugs. And what it does is it enables this rapid testing with demonstrated safety profiles for new indications. And this is the library that was used in this landmark drug repurposing screen for COVID-19 by the Scripps Research Institute. And, I won't delve into too many of the experimental details here in the interest of time, but, as we had sort of discussed earlier on, you have a compound library, you have concentrations or doses that you're testing these compounds in, and you have a cellular system. Now, again, one of the arguments to be made for having a platform in place is so that we have biologically relevant cells that you're testing in. So I will say right off the bat, having dealt with the Vero E6 cell line, it is not necessarily indicative of disease, but one of the biggest sort of problems that we do have typically is for a lot of infectious diseases is that they're BSL-3 agents, and you can't actually, for many labs, it's impossible to sort of have a BSL-3 facility to do high throughput testing. And so we use proxy models in those cases. And this is an example of a proxy model where you have the infection with SARS-CoV-2, and then you look at cytotoxicity or cytostatic effect in the cellular assay of choice. And what you can see over here is that they actually, looking at the FDA-approved drugs and the preclinical compounds, they did actually find small molecules that had already been approved and sort of fell into these different classes, whether you're looking at cysteine proteases and antimicrobials, drugs that were hitting the ion channel, G protein coupled receptors. They did test some chloroquine derivatives, but as you can see, they had multiple hits within these classes that were actually significantly reducing infectivity or not only infectivity, but causing cell death of cells that were sort of the virus within the cellular system. And now that these have sort of been published on, the idea really would be is for a lot of the approved compounds to be able to be taken into a clinical setting in a very controlled manner and cross-referenced against the comorbidities of the patient to be able to see, for example, with a diabetic patient, what should you be giving? You probably don't want to give a G protein coupled receptor drug to a diabetic patient, but an antimicrobial would work well, a cysteine protease would work well. For a patient that already has cardiac issues, you could perhaps give ion channel derivatives or G protein coupled receptors. So that was, what this has done. This drug repurposing screen has been to really sort of change the way that we treat individual patients, because we now know retrospectively after 7 months that a lot of the severity of this particular disease depends on the patient. And so to be able to tailor to the best of our ability in this short period of time to different populations of patients becomes very important. And, I'll sort of end with the different phenotypes that we're talking about. We've talked about pathology. We've talked about the organ-specific sort of targeting of these drugs. But what are the phenotypes that we're talking about? And if you notice in the previous slide where we talked about the different pathways that we were targeting within a cell, but what does that mean in terms of viral infection? So what it means in terms of viral infection is when a virus actually enters the cell, where are these different receptors that we're targeting that are actually seem to be efficacious in reducing viral load? What are the different opportunities for therapeutic intervention from a biological perspective, and how does that sort of how are we able to marry that into bigger sort of, looking at classes of therapeutic agents as a whole. So again, here it comes back to thinking about clinical research as a portion of basic research, or vice versa, where you're looking from that macro organ systemic perspective right down to the cellular level to be able to see how we best tackle this disease from the process, from the cellular processes up to the comorbidities and patient health at the time of presentation of disease. So what it means for us as a community, both for scientists as well as clinicians, is being able to understand the role of big data in translational medicine. And this is an idea that I would like to end with, because what it really sort of tells us as a community is that we have to be able to look at data from different sources. We have to be able to combine data from different sources. So with COVID-19, as I'd mentioned earlier on, with the drug repurposing, a huge component is looking at in silico data, because we don't have the cellular systems, we don't have the patient population to mirror. We do have some clinical trials. Again, we don't have, when these are not very stringent because of the ethical considerations involved in being able to treat patients who are, affected by COVID-19. There are in vivo studies, there are animal models biometric data that's being sort of generated right now. We do have a lot of cell-based in vitro data, as I mentioned earlier on. There is genomic data now available post-mortem from a lot of patients that have been affected. There is now an effort being made by rare and orphan disease foundations to collate this data to sort of help build platforms of the future. So what it means for us really, if we want to be able to develop these predictive paradigms for any future sort of public health scenario, be it an infectious disease, be it a biological hazard, is really being able to understand the different sorts of data across the board to be able to use our patient data much, much more meaningfully in the context of a research setting, so that when push comes to shove, when we all have to mobilize to be able to quickly turn around therapeutics, we have all the data, we understand the data, we understand how the data interacts with each other, and we're able to, sort of hopefully mitigate the disasters that we're sort of seeing right now with COVID-19. So with that, I would like to sort of end this particular section, and thank you for your attention, and I am happy to take any questions that you might have.
Thank you, Dr. Madhu, for that very lucid presentation. We indeed have some questions. First and foremost, looking at your models and the way in which so much of information and data and trying out almost more than 1 million molecules to find one drug. So that reminds me of this quote, no? That science is only 1% inspiration, 99% hard work. You need to eliminate all the possibilities before we get it right. Yeah, that is, having said that, and then there are a few questions which have come from the audience. The first and foremost question is from Dr. Bhatt. Yeah, so he is saying that many infected people in this corona pandemic are asymptomatic. Can we stop sharing, please? Yeah,
I can stop there. Yeah.
Yeah. So many of the people are asymptomatic, especially. So if we are going to give them the antiviral drugs, will it decrease, one second, let me get, yeah. Will it reduce the rate of transmission of the disease? That's the first question what we have from Dr. Bhatt.
That's an excellent question. When I talk about sort of the retrospective data that we've been seeing, a lot of it has been post-infection. So we understand, how to treat and what the manifestations are post-COVID-19 infection. With asymptomatic patients, there seems to be a window of when viral replication is taking place. So there are some studies going on right now where you're looking not at the early replication phase when, within the first 5 to 7 days of exposure to the pathogen, but much later on, just before the viral shedding, between 12 to 14 days. It seems like at that late stage of viral replication, when, it's sort of your viral load is sort of exponentially increased, treating with an antiviral at that particular time would actually be efficacious in terms of reducing the severity of sort of transmission. And that, again, to your point, and I think that's an excellent question that was brought up, that's where having a robust process in place for contact tracing and being able to sort of predict people who might be asymptomatic transmitters would be hugely beneficial.
But the only issue treating asymptomatic patients, especially in a country like India, many a times we may not even be able to identify them. That is one thing, and the cost. Right. Who is bearing the cost for the entire thing?
Yeah.
Okay. Yeah. And there is one more question from Dr. Wasim. He would like to ask, what is your opinion about the vaccines which are expected to be launched earlier than expected, as per the reports which are emerging from certain countries? Quite a every day they keep saying tomorrow it will come, day after it will come. That is how it is going on.
So unfortunately, I cannot comment on that because the vaccines are being regulated by the FDA. So that is, I would love to be able to speak on that, but at this point, I can't comment on that because it is regulated by the FDA, and that commentary would have to come officially.
Okay. And there's one more question. Okay. The other one is doing clinical trials on a large scale in patients who are asymptomatic. This is from Dr. Muhil from Trichy SRM. So she would like to know about how we can do clinical COVID-19 clinical trials for newer drugs on asymptomatic patients on a large scale. Is that acceptable? So that is the question.
That's an excellent question, because there has been, we talked about the ethical considerations with the placebo arm for people who are actually sick. So, that's really not an option for people who are actually presenting with the disease. But then, there have been so many clinical trials that, are ongoing right now all across the world, but we don't have, quote unquote, that control arm to be able to, sort of, there's no basis for comparison. It's all a very sliding scale of the least affected patient to the most affected patient versus disease and normal state. So there is a lot of talk about, perhaps we make the sample size much smaller, but that normal arm would actually be your asymptomatic patient, because what that would end up doing is being able to give us a better assessment of how these drugs actually affect disease severity in patients who are actually presenting with it. But also, to go back to that previous question, it would give us a good sense of, for whatever class of drugs that we're looking at, in an asymptomatic population, are you able to stem that, viral replication early on so that these asymptomatic patients don't spread?
Okay. Thank you. There's one more thing which is from my end. Like when we are talking about systems approach or systems thinking for finding solutions to problems, we always keep thinking, or we always keep talking about find your last pilots, right? Always there has been somebody who has attempted it and it is there somewhere in your system. Right. I think what you spoke about drug repurposing, especially looking at it at molecular level and trying to identify those, is trying to look for our last pilots. That is understood out of it. Yeah. Okay. I think there are no further questions. However, there are quite a few comments which are there in the chat box. Dr. Manoj Moses wants to congratulate you for a very lucid and clear presentation. Very well presented. That's what they say. Yeah.
I
Any
appreciate
words
that. Thank
from?
you.
Okay. I thought Dr. Vijay will have some questions, but that doesn't look like ma'am has any questions. Any words from you, Dean sir? Madam, do you want to say something? Okay. I've unmuted you. Do you want to say something, Dr. Vijay? No, I don't think so. Okay. So I think there are no further questions. Thank you on behalf of the Scientific Society of AVMC and Vinayaka Mission Research Foundation. I thank you, Dr. Madhu, for an excellent and lucid presentation on a quite a difficult topic, and for an audience who's clearly this is a new approach to do with. So you made that transition quite easy, and especially your slides were very interesting. Right. Okay. Some other question has come. Okay. So this is with regard to the talk. Thank you very much for your time, and thank you for sharing this. I would request your permission to share the slides with the audience, because when we
Absolutely.
Yeah. Thank you very much. So the audience can get their slides. Yeah. And
absolutely. My pleasure.
Thank you. And before we conclude, we have some housekeeping messages for the audience. First and foremost is your e-certificates for this program will be available. You will get an SMS saying that please get this information. You can look at the, you can go to the site and download the certificates. Only thing is you need to use the ID which you are using to log into Cisco. Suppose if you are using any other email ID, it will not recognize. It is indexed to your Cisco ID which you use to participate in this program. So that is the regarding the e-certificates. And we have another interesting talk scheduled next week. We'll be taking from this and we'll move a little further. We're going to talk about how genetics is going to influence clinical medicine and what we can do. So we have Dr. Girisha, who is from Manipal, who is the professor of pediatrics in Manipal, and he is also running the human genetics unit there. So he will be talking to us about genetics and its influence for clinical medicine implications. So that is for next week. On behalf of the Scientific Society, this is Dr. Mahalakshmi, Dean, Health Professions Education from AVMC, signing off. We would meet you again next week. Thank you. Stay safe, folks. Bye. Bye, Dr. Madhu.
Bye. Bye. Thank you.